Log on, tune in, find out. Another good idea from Cambridge.
Well, a very warm welcome to this 27th Darwin College lecture series. As you'll, I'm afraid it's wetter inside than in tonight, so hence the blankets. For some reason, the Lady Mitchell Hall is leaking. As you'll be aware, the theme this year is life. And as usual, we've got speakers from 8 very different disciplines giving their perspective on the idea of life and its limits and its boundaries. But less usual, you'll have probably noticed from the programme that very few of our speakers are going to have to travel far to get here. Most are from Cambridge. Indeed, actually, most of them are from Darwin College. However, this isn't the result of parsimony arising from the financial crisis. Quite the contrary. Once again, we're deeply grateful to Richard and Anne King for their generosity in supporting the series. The reason for this apparent parochialism is quite different. In many ways, it's an indulgence. This is the 12th and last series which I shall introduce as Master of the College. And Andy Fabian and myself, who put together the programme, thought that it was a shame not to take advantage of the fact that there are in Cambridge and quite close by some of the outstanding speakers and communicators about their subjects, some of whom, alas, you're more likely to see on television than to hear give a lecture. So we thought we'd have the home team, and I'm very confident that they will be outstandingly good. Now, it's appropriate for a Darwin College lecture series to start a series on life with an exploration of life's evolution. And tonight we've got Michael Akam, and the title of his speech his talk is From Genomes to the Diversity of Life. Michael is the Professor of Zoology, and he was previously Director of the Museum of Zoology here in Cambridge. He was trained in Cambridge and in Oxford. And his research has been concerned with shedding light on the genetic mechanisms that control animal development. He started with nematode worms and fruit flies, and he's moved on to the genetic controls of arthropods, which to most of us are the wiggly things that have segmented bodies and jointed legs. And currently he's focusing on centipedes. Michael's path-breaking research has been honoured in many ways, most recently with a medal from the Linnaean Society. It's a great pleasure to introduce Michael to talk about From Genomes to the Diversity of Life.
It's a great pleasure to be here giving the first of this year's Darwin Lectures. Our genomes are the complete set of genetic instructions that we carry within us. For each of us, there is a single large DNA molecule packaged into each of our chromosomes. And those chromosomes, copies of those chromosomes, are packaged into the nucleus of every cell. There are a few little bits of the genome that lie outside the nucleus. I'm going to ignore those today. This genome carries the coded instructions to make, directly or indirectly, all of the large and complex molecules that make up our body. And our own genome, together with indirect contributions from the genomes of our parents and our parents' parents and so on back through many generations, provides all of the information, all of the heritable information that allows those components to be assembled into a body during our development, that specifies how that developing body will interact with the environment to alter itself as it matures, and then how the fully adult organism, animal, will behave and reproduce throughout life. So genomes are important, and studying genomes is interesting. I think sometimes people think that the genome is the only level of biological organization that it is interesting to study at the moment. And that, I think, is a very wrong view. You cannot explain all of biology just by looking at genomes any more than you can explain the weather by thinking about the quantum behavior of atoms. But genomes do have remarkable things to tell us, and I want to explore just a very few of those things today. I am going to leave out huge areas of biology. In particular, I'm not really going to talk about what we have learnt from the human genome sequence about medical issues or indeed the nature of man himself. I do think, though, that if I were starting science again, I would probably become a genetic archaeologist and try and use that combination of history and function that is written in the genome to understand how the diversity of man has become as he is today. I think that is a fascinating subject. But I'm a zoologist. I know more about obscure animals, and I'm going to start with that area of biology. And I apologize to all those here who want me to talk about plants, or indeed the diversity of bacteria and viruses. It's all terribly interesting, but I don't know very much about it, and I'm going to say almost nothing about it. In fact, I'm going to talk about almost nothing but the genomes of that group of organisms that have cells with nuclei, the eukaryotes. It's still quite a good amount of diversity, as I hope to show you. The history of genome sequencing has been an extraordinary success story over the last 25 years. In 1979, as a newly finished PhD student, I was privileged to spend a few months at the Laboratory of Molecular Biology, where Fred Sanger had demonstrated rapid techniques, relatively rapid techniques, for sequencing DNA just a few years previously. And I was part of a group of 5 or 6 of us, who worked for a couple of years to sequence the genome of the tobacco mosaic virus. It's about 6,000 bases long. We were very proud of completely sequencing that genome. The largest genome sequencing center today, the BGI in Shenzhen in China, can produce 5 terabases of raw sequence data a day. That is 1,000 times as much DNA as is written in the human genome. In fact, you're probably very familiar with Moore's law, which indicates how the power of computer processing has advanced in recent years. Well, the white line at the top of that curve is how much DNA sequencing would be improving if it was improving as fast as computers. And the actual line is the blue line, plotted from 2001 to 2011. And what you can see is that something dramatic happened in 2008, when what's called next-generation sequencing dramatically dropped the cost of reading DNA sequences. This is a technology that allows sequences to be produced in an automated, highly parallel way that really completely changed perspectives on what one could think of doing with sequencing. In fact, it is probably true to say now that the simple raw cost of sequencing DNA is not going to be rate limiting for any biological project. Virtually any organism that we are interested in, we can sequence the genome. Assembling that genome, taking the tiny fragments that come out of the sequencing machines and turning them into an organized array of bases, A, G, C, T, and so on, is still challenging. And reading the information that is encoded in those genomes is intensely challenging. But the basic raw cost of reading genomes has now dropped to the point where we are told that next year we should be, no, this year, 2012, we should be able to read a human genome for about $1,000 in about 2 hours. It's a complete revolution, really, in biology. I want to talk about the impact of all of that genome sequence in a number of areas. And the first one I want to talk about is its impact on our understanding of the pattern of evolution particularly of the animals. From Darwin's very first sketches of trees of relatedness of animals, it didn't take very long before biologists were beginning to draw really rather complicated trees depicting how all of the organisms that we are familiar with might be related. Ernst Haeckel drew this really rather splendid tree, and like most of the biological trees that were produced in the last century, up until the later part of the last century, it combined a mixture of real insight and speculation and prejudice. For example, man is right at the top of the tree, and by and large, as you go up the tree, things become more complex. And you may not be able to see it, but near the bottom of the tree, that bottom right-hand twig is something called infusoria, the single-celled organisms that appear in ponds and swim around in the water. As you will see, our perspective on the diversity of life has changed quite a lot. It was in 1988 that the first major paper purporting to tell us the relationships of the animals, the phylogeny of the animal kingdom, was published as a high-profile paper in Science. I remember it coming out. I remember being very excited by it. This paper was based on the sequence of one gene that is present in all eukaryotes. It's the gene for ribosomal RNA, about 2,000 bases of that coded sequence. And because of developments in sequencing techniques, it had been possible to read the sequence of that gene in a relatively large number of different species, 22 classes in 10 of the different major groups of animals. The sequence is not only a coded set of instructions, but it is also a record of history. There are many changes that happen to the genome, changes of single bases from a C base to a T base, for example, which may have relatively little significance, but which just represent noise in the process of copying DNA. And changes like that accumulate over time and are rather powerful for recording the history of genomes. And if genomes share many of those changes, they presumably come from closely related organisms. And then there are more complex changes, and I'll show you examples of other changes where the order of genes change, for example, which also record history. So molecular sequences provide us with a way of looking at the history of life. What is most striking about this paper is that almost all of the major conclusions that were published would now be regarded as wrong. The classical eumetazoa, that means the animals, are divided into two groups. Cnidarians, the sea anemones and so on, arose from a protist, one of these single-celled organisms, protist ancestry different from the second group, the Bilateria. A new and radical conclusion, completely wrong. And actually everything else in that abstract is probably not correct. So if this was an approach to biology that had its teething problems, but actually in the 20, 25 years since then, our ability to understand the processes that generate those historical changes, and then to infer from the pattern of those changes across many different animals, the relationships of the animals, has improved enormously. And although I am sure that there are things, in fact I know that there are things on this slide that are wrong, and this was one of the more recent publications, this is now based not on a single gene, but on the sequences of a large fraction of the genes, or a significant fraction of the genes, in whole genomes. And it's providing us with a pattern of relationship that is much more robust and, in some respects, very surprising, almost as surprising as those early erroneous conclusions. The first thing I would just like to mention on this is our place in the tree of life. We are not, in this wonderful diagram produced by Giselle Walker, who was a fellow of Darwin particularly interested in the infusoria, in the protists. We are not at the top of the tree. Turns out that the real diversity of life lies among those infusoria. The different lineages of single-celled nucleated organisms have been evolving for perhaps 2,000 million years, and they are extraordinarily diverse. And if you look down in the bottom left-hand corner of that slide, you will see that all the animals are represented by a fly and a starfish. They are but one twig in this bush of life. And that really, the relationship of these different single-celled forms and the multicellular forms that came from them is something that has only become clear since we've been able to sequence genomes of many of these organisms. In fact, 10 years ago, when I began to teach lectures on animal diversity, I had no idea where in that bush of genetic lineages animals arose. There were ideas that they came from ciliates. I don't seem to have a pointer, no matter. There were ideas that they came from some of the animals in blue, or the organisms in blue. There were ideas that they came from amoebae. But we now have a very good idea of the lineage of single-celled creatures that is at least our closest relative among those that have been studied so far. And it turns out that it was a very old idea that was correct. A man called Saville Kent, working in the 1880s, had noticed that one lineage of those single-celled organisms, so-called choanoflagellates, they're these rather delightful cells shown in panels A and B there, that have a single flagellum that makes a current of water, and then a collar around it of very fine tentacles, which filters particles out of that column of water and allows the cell to absorb them to feed. He noticed that those choanoflagellate filtering cells were extraordinarily similar to filtering cells in sponges. You may not think of sponges as animals, but they were recognized long ago as sharing many characteristics with other animals, and are confirmed as one of the basal branches, one of the earliest diverging branches of the tree of life. For about 100 years after this hypothesis was put forward, it was simply one of many competing ideas about the origin of animals. But one of these choanoflagellates, the genome was sequenced. It's an organism called Monoceca brevis, sequenced in 2008. Sequenced, I might say, because there were a number of hints from earlier studies that Saville Kent might have been right. And it is now very clear that he was right. This single-celled organism swimming in the sea, and these are quite abundant organisms, shares many genes uniquely with the rest of the animals that are not found anywhere else in that great diversity of forms. So we would guess that the last common ancestor of all of the animals with a unicellular form might have looked something like one of these choanoflagellates. And it turns out that choanoflagellates are actually rather good at switching from being single cells to being small groups of cells. When, under certain feeding conditions, it's better to be a large than a small unit, they stick together and they move around as these clumps. And so this might be a model for the type of organism that gave rise to all of the rest of the animals. For obvious reasons, the genome of these choanoflagellates has become a very interesting place to look for the origins of what allowed us to become animals. I'm going to go right to the opposite end of the phylogenetic spectrum, the tree of animals, if you like, just to illustrate one other aspect of the diversity of animals where genome sequences have helped us. 3 pictures of animals on the slide. A European mole, sort of animal that might pop up in the middle of your lawn. A Cape golden mole, which might pop up in the middle of the garden if you lived in South Africa. And an African elephant. Obviously, I wouldn't have put that slide up if the obvious pattern of relationship was true. And I'm cheating a bit, because it's been known for a very long time that golden moles are not actually close relatives of our European moles, but are something else. If you look at the details of their skull and their structure, comparative morphology recognized that they were something different. But, of course, convergence has been very powerful in restructuring the body, in making those 2 animals with rather similar lifestyles look the same. And that made it extremely difficult to figure out where they should go in the tree of the mammals. In fact, it is the golden mole and the elephant that are the closer relatives. Sequencing the genome of mammals in the last 10 to 15 years has revealed that there is a group of species that share a large number of genomic characteristics that identifies them as being more closely related to one another than they are to any of the other animals. And they don't look very similar. The golden mole is a relative of the tenrex there. You can see the aardvarks, the elephant shrew, which only superficially looks like an elephant and is a mouse-sized creature rather than an elephant-sized creature. Dugongs, manatees, and so on. Many of these were organisms that had been, or species that had been hard to place, but I don't think before the molecular sequences began to arrive, anybody put all of them together in the same group. My colleague Rob Asher in the Museum of Zoology is particularly interested in this group and characteristics that they share. And what I think is now almost universally accepted is that this group of animals represents a single lineage that are all more closely related to one another. And I've just put in one piece of evidence of the sort of evidence that shows this. In our genomes, as well as the useful genes that encode parts of our own structure, there are also sequences that are there largely for their own purposes, to copy themselves and make extra copies of themselves in the genome. And these genes can jump from site to site in the genome and insert into new positions. When a gene jumps into a new position, it's quite likely to stay there and not leave, and that gives you a signature in the DNA that all the descendants of the ancestor where that happened will share. And this is a little bit of the sequence. You won't be able to read it at the bottom, but you can see that all those mammals at the top have basically no sequence in a particular place where all those Afrotherian mammals share the insertion of a sequence. It's many of these rare genomic events, the insertions of jumping genes or other reorganizations of the order of genes on the genome, which provide additional information as well as just the sequence of bases to indicate that those mammals really are more closely related to one another than anything else. What that suggests is that geography is actually much more important in the history of the evolution of mammals than perhaps was previously recognized, because all of those lineages appear to be lineages that diversified early on in Africa and that's the reason why they've been called these Afrotherian mammals. This is just one example where dramatic radiation and diversification of species has made it very difficult to see what their ancestry is, but there is sufficient information in the genome to tease that history apart. I could go on for this lecture and many more about the things we've learned about the relationship of animals, but let me move on to what is in a genome. There are all sorts of information in genomes, but when you sequence a genome, this is actually a little bit of the sequence of my favorite centipede genome. When you sequence a genome, it doesn't leap out at you. This is the first few hundred bases, and this particular fragment that came out of the assembly of our sequence goes on for another 500,000 bases, and it would all look pretty much the same. It's actually a very interesting part of the genome, but just looking at the bases is not enough. You need a computer to look at those bases and try to interpret it. And there is an industry of people who are spending their lives finding how to generate and display data from genome sequences in ways that is useful. For example, you can find which bits of the genome are translated into messages. You can find which bits of the genome look like bits of other genomes where we do know what they do. You can find which bits of the genome stay the same in closely related species rather than diverging quickly. And in this genome browser, all those different sorts of evidence about how the function of that DNA might work can be depicted in an enormously condensed version. This is a picture of the whole 500,000 bases of that particular fragment. And then the computer can make guesses about what that evidence means. And the guesses are those funny little diagrams in the middle of the picture which indicate where it thinks there are genes. The little arrows between bits are where it thinks there is a bit of gene that is an intron, and the squares are where it thinks there's a bit of gene that is coding for message. And it's really a depiction of gene structure. And with these wonderful programs, you can zoom in and blow up until you actually see the final sequence of bases. But rather to my surprise, I mean, I'm a novice in this business of trying to read genomes. Rather to my surprise, it is still quite difficult just to figure out how many genes there are in a genome. Certainly, these computer programs are not perfect at it. It's quite easy to find bits of genes that code proteins. We know the translation code from DNA to a protein, and therefore you can say, this bit of the genome makes a component of hair, or this bit of genome makes a component of muscle, the myosin protein. But for proteins that are not so well understood or so well conserved, or where the gene is complicated, guessing how many genes there are is still challenging. So in fact, our understanding of what genes are in genomes is still not terribly good. There's quite an interesting history here. Before we had the sequence of any mammalian genome, the general guess from geneticists was that fruit flies might have 5,000 genes, and mice, mammals, humans might have 100,000 genes, 20 times as many. In fact, when I was thinking about this, I seem to remember discussing this with Martin Evans 20 years ago. Martin was then my colleague in the Wellcome CRC Institute. Since then, he's won the Nobel Prize for discovering mouse stem cells, and now he's in Cardiff. But in the time-honored tradition of arguing about what might be true, we had a bet as to how many genes there are in the mouse. I thought that it would actually be more than 100,000 genes to make a mouse. I was very familiar with the fruit fly data, and I thought that a human being was a lot more complicated than a fruit fly. Martin at that time was not so sure. He thought it would be less than 100,000. I have to say Martin was absolutely right, and I have never bought him a fine walking stick, which I owe him. But anyway. So when genomes were sequenced, and this is a picture from 2002, there was a comforting trend in the size of genomes as animals, as organisms became more complex. I've actually got some bacteria on this slide. So bacteria, single-celled infusoria like yeast, baker's yeast, for example, 5,000 genes would be a good guess. Fruit flies, nematode worms, Caenorhabditis elegans, the worm that Sidney Brenner started with, 10 to 20,000 genes seem to be the sort of estimates that were coming from the first analyses of the genome. And the early analyses of mammals, mice, humans, suggested not as many as 100,000, but perhaps 40,000 genes that made proteins. Actually, that's been revised downwards quite substantially. The genome programs were probably not recognizing how genome analysis programs were not recognizing just how big some of the genes were, and that the actual correct number was rather smaller than that. And in fact, the difference in gene number between humans and fruit flies is now disturbingly small. Indeed, you probably don't have as many protein-coding genes as that little weed, Arabidopsis, in your body, which I think surprised most biologists. Gene numbers are not a very good indicator of complexity of organisms. This genome of the sea anemone Nematostella was sequenced in 2007, a few years ago, and that made that problem even more acute. This sea anemone is believed to be a representative of one of the very early branches of the animal kingdom. This is a simplified version of that complicated phylogenomic tree. Everything that you probably think of as a proper animal is in those green, blue, and red boxes on the left, all the worms and flies and snails and fishes and so on. And then there's a few groups that appear to be deeper branches, earlier emerging bits of the tree of life. The very deepest is a sponge, and it looks as if actually the very earliest radiation of the lineage that gave rise to us was a radiation that gave rise to several different lineages of sponges. But the sea anemones belong to a group that diverged from the rest of us very early. So any complexity in the genome of a sea anemone that is shared with the complexity of those so-called bilaterian animals, the more complex animals, represents complexity that had already evolved right down at the bottom of that tree, at the beginning of the radiation of the animals. I'm sorry, this is a very complicated slide, but let me just talk through it very briefly. It's a slide that plots the number of genes of a particular gene family called Wnt genes in the genome. You don't have to know what a Wnt gene does, save that the Wnt genes encode signaling molecules that are absolutely critical for almost all aspects of development, for the organization of your brain, for the elongation of your vertebral column, for the patterning of almost every organ in the body. Without Wnts, you would not get very far in embryogenesis. There are Wnt genes in all the animals that have been looked at, but at the time when this slide was made, there were far more Wnt genes in humans than there were in, say, fly or worm genomes. Those crosses mean genes that had been found in human but had not been found in flies or nematode worms. And the interpretation of that slide was that that meant that the humans had invented all these new genes to help them become more complex. And then that sea anemone genome was sequenced, and it turned out that an awful lot of the genes that were missing in flies and worms were actually already present in the sea anemone. And the conclusion from that is that actually flies and worms have been streamlining their genome. They've been throwing genes away, but that the early common ancestor that we share with the sea anemone had already invented, if not every single copy of a Wnt gene that is already in the human genome, at least 9 out of the 10 different families, recognizably different types of this Wnt signaling molecule. That surprised everybody. What on earth is this animal that has just a few cell types and very little complexity doing with all those genes? we really don't know. Another story of complexity, slightly different, but I think I might just have time to tell it. This is the picture of a fossil, Pikaea, which is a representative of our own lineage, the chordate lineage, that flourished in the Cambrian oceans. And the Cambrian is the geological period when animal fossils first appear. And the prettier picture is a reconstruction of what that creature may have looked like. 600 or so million years ago. Luckily, for geneticists, creatures remarkably similar to this are still alive today. There's an animal called Amphioxus that sits in the sands in Florida most of its life, sucking particles out of the water, 5 centimeters or so long, which looks remarkably similar to that Cambrian animal. It's not a vertebrate, it has no vertebral column, but it has many of the features that we recognize as characteristic of vertebrates. And for that reason, zoologists in the 19th century thought that this was a relative of ours, and they called it a chordate, cephalochordate, and we are vertebrate chordates. When that genome was sequenced, it did indeed turn out to be one of our closer relatives among the animals. So this little tree shows you Amphioxus and a representative of the vertebrates, Charles Goodrich, who was an Oxford zoologist. But it's a little disturbing because actually the genome sequences suggest that the closer relative of Charles Goodrich is not Amphioxus, but a sea squirt. Sea squirts were sequenced in 2002. They too had been recognized by 19th century biologists as probably related to our own lineage, not because of any characteristic of the adult. The adult is basically just a sac that sits. If you go down to Plymouth Marine Station on the jetties there, you can feel them as sort of nasty jelly growing on the pilings that support the jetty. So the adult doesn't betray very much of its ancestry, but turns out that these creatures have a larval form that is much, much more like vertebrates, something quite extraordinarily like a tadpole, and that had made them interesting. But because of the enormous difference between the adults of the sea squirts and the vertebrates, it had been generally assumed that they were a fairly distant relative of the vertebrates. Alas, the genomes, after some controversy in interpretation, are now suggesting very strongly that actually the tunicates the sea squirts are our closer relatives, and in fact that they must have lost many of the characteristics that we share with that Cambrian vertebrate-like Amphioxus. And this tree has now got plotted onto it representatives of a family of genes that I've spent a lot of my career working with, so-called homeobox genes, which are important developmental control genes. They're genes that tell cells what type of cell they should be, and they tell animals where they should, they tell cells where they should be in the body. They're the sort of genes we expect to be crucial for building bodies. And what the genome of the sea squirt told us was that sea squirts have actually thrown away a lot of those genes that they inherited from the last common ancestor. There's just many genes that we can infer by comparing Amphioxus and humans and sea anemones must have been present in the common ancestor that have simply been thrown away as the animal became much more simple. So genomes become more complex, but they become more simple as well. One of the ways in which animals became more complex when they became multicellular was to invent new cell types. Single-celled animals, it's not that they're simple, they are quite extraordinarily, single-celled organisms. They are quite extraordinarily complex cells, but individual cells do everything. And one of the important things about becoming a complex organism is that you specialize. You have nerves, you have blood, you have muscle. I just want to say a few words about a recent study of how the diversity of cell types might have arisen that has been informed by genome studies. It concerns this particular animal. It's a worm. It doesn't actually look anything like as pretty as that lithograph, unfortunately. The colours, I think, are optimistic. But it's a marine worm, the sort of creature that if you're a fisherman, you might be digging out of a muddy beach to use as bait. We think of that as a simple animal, but actually it is quite complicated. But this marine worm has a larva that is wonderfully simple. It's a swimming larva that has cells with beating flagellae around the side, or cilia, that enable it to move around in the water column in the sea until it settles down and transforms, metamorphoses into a worm. And that larva has become a model for trying to understand the origin of cell types, partly because it is so simple. And the nervous system that enables that animal to swim is pretty much as simple as a nervous system can be. This is a picture, a section through that larva. You can see the ciliated cells around the outside. You can see one nerve that connects up all the swimming cells, and you can see just a couple of neurons, 4, 5 neurons in the middle. That's all it has by way of a nervous system at that stage in development. It will become much more complex. But it's enough to behave, to show the first signs of complexity, except that I don't seem to... there it is. So this is a picture of a plate full of those larvae in, if not the dark, at least with uniform lighting. And I'm going to play the movie, and the light will turn on when it blinks. I think I'm going to play the movie. Yes, there we go. They're swimming around doing nothing, and then there's a blink about now, and the light has been turned on, and that very, very simple nervous system is quite good at changing their behavior. How does that work? Turns out that it is a beautiful model for the origin of diversity among cell types. Detlef Arendt and his colleagues at Heidelberg have shown that that whole system depends on an eye that is made up of 2 cells, one photoreceptor that actually senses the light, and one shading pigment cell that makes that photoreceptor directionally sensitive. And then those photoreceptors directly interact with the ciliated swimming cells, and they change the beat of the cells such that, depending on where the light is, the animal will actually spirally move towards it. It's a pattern of behavior that is seen in many of these small marine larvae. It's also seen in many of the single-celled organisms that also swim and move towards light. And he's been very interested in finding what genes specify this diversity of cells, because he's proposed a really rather nice hypothesis for how that diversity arose. And it's basically a hypothesis that all our different cell types arose by a division of labor from initially much more complex cells, perhaps not the way we normally think of evolution. Let me just go through this a little bit. If you look at a flagellate photosynthetic unicellular organism that needs to swim to the light, or indeed if you look at the photoreceptive cells in a box jellyfish, the cell that senses the light also contains pigment and also has the swimming flagellum, and it is the direct effect of the light on that cell that changes the way that cell itself drives the swimming behavior. So ancestrally, we suspect, or Detlef suspects, that there were multifunctional cells, and that this first step towards making a much more complex organism, the first step to building a nervous system, actually involved splitting that complexity such that each of the separate cells could then become much more specialized. And the reason he thinks that is that looking at the set of genes expressed in the photoreceptor cell and the shading cell, for example, it turns out that they share many of the critical control genes that specify cell type, those homeobox genes that I mentioned earlier, and many other sorts. In fact, in terms of a molecular fingerprint, they are more similar to one another than either is to the photoreceptor cell or the shading cell in the adult eye. And he suggests that this is evidence that that complexity of cell types has actually arisen by what you can think of as a tree of the origin of cell types, analogous to the tree of the origin of species or the tree of the origin of genomes, but that we can begin with this molecular fingerprinting to actually plot how the diversity of cell types arose. I don't have time to go into that in more detail, but the extraordinary thing is that by collecting these molecular fingerprints, these sets of genes that are expressed in particular cell types, he is beginning to be able to draw comparisons between the cells of this annelid worm and the cells in vertebrates, and to suggest that both the vertebrate nervous system, our own nervous system, and the worm nervous system are actually built around a set of rather conserved units where we can still read, in the way the genes are expressed, the history of how some of those cell types have changed and diversified. I think this is very exciting. It's still quite speculative. There are some people who don't believe it, but I think that this idea of understanding how the complexity of cells in our body have arisen is a very interesting new way that we can use molecular signals. But just making cells different from one another is not sufficient to make organisms. You not only have to make cells, but you have to put them in the right place. Some animal species are quite extraordinarily good at putting cells precisely in the right place. It was realized in the 1890s that if you looked at snail embryos and you looked from individual to individual, you could see that the pattern of cells in the cleaving embryo was exactly the same. So constant, in fact, that you could give a specific name and number to every cell, and you could indicate which of the cells in the early cleaving embryo would give rise to which cells in the later embryo. And indeed, not only could you predict where the cell had come from, but by looking at the middle stages, you could predict what that cell would become. A particular cell, say the cells marked in red in that middle diagram, would go on to make particular muscles or particular skeleton structures or whatever. Putting cells in the right place is a critical invention of being a complex animal. It's really the invention of embryology. Now, this particular lineage of animals, the snails and annelid worms and their relatives, have taken it to an extreme. We are nothing like as careful as that when we make our own embryos. But all animals have to pattern their embryos somehow so that cells know where they ought to be, whether they're part of the head or part of the tail. And one of the surprising stories that came out of early comparisons of how genes are used in building embryos is that there is much more in common than we might think in organisms that appear to be radically different. This is an old story, but I've chosen a slightly new slant on it. I've chosen to illustrate it with a comparison of vertebrates, ourselves, mice, fish, and acorn worms. Now, you may not be familiar with acorn worms. Again, they're another of these marine groups that burrow in the sand, but they don't look much like us. And if you wanted to put a coordinate system on the embryo of an acorn worm, this is the embryo of an acorn worm, developing embryo, you might be quite hard-pressed to say how you would compare that animal with a developing fish or mouse or human. But when you look at that critical set of genes that control the specification of the embryo, these transcription factors, genes that tell other genes to turn on and off to make particular sorts of cells, it turns out that there is a quite extraordinary degree of similarity in the early patterning of this acorn worm and a typical vertebrate. And vertebrates are all rather similar at this early stage in development. So you don't need to read any of the labels, but the colored bars indicate the domains of the embryo where a particular gene is expressed. The head end of a vertebrate expresses a subset of genes, and it is those same genes that are expressed in that proboscis of the acorn worm. And then there's the midbrain region of us, which expresses a different set of genes in the early embryo, and that collar region of the acorn worm expresses the same genes. And then in the back end, there's the famous Hox genes, which tell our neck vertebrae to be different from our chest vertebrae, to be different from our lumbar vertebrae. And the same set of genes expressed in a similar array of domains is doing something in the very boring back end of that worm. We don't know what it's doing, but underlying that diversity of form, there is some sort of conserved coordinate system that seems to be present. Actually, not just in these two representatives of one lineage, but it's largely preserved across all of the animals. I could have drawn similar pictures with a fruit fly, for example. In fact, that's true for the anterior-posterior axis. Very similar sorts of comparison suggest that another very old idea is true for our dorsal-ventral, our back-to-belly axis. Geoffroy Saint-Hilaire, famous French biologist of the 19th century, looked at vertebrates, and he thought that there was perhaps a relationship between the organization of the vertebrate body plan and the organization of the lower animals, as he would probably have called them. For example, vertebrates have a dorsal nerve cord in green and a ventral aorta in red. And all you have to do is to turn them over, and you get an animal with a ventral nerve cord in green and a dorsal aorta in red, which looks remarkably like a typical arthropod. So he actually proposed that vertebrates and invertebrates, or many invertebrates, were sharing a common ancestral organization, but that there had been an inversion of our back-to-belly axis. Cuvier, Baron Cuvier, who was the most powerful zoologist of the time, absolutely trashed the argument. I mean, he rubbished Geoffroy Saint-Hilaire, and the idea died. But it was resurrected repeatedly over the next 150 years. And it now looks, from exactly the same sort of data I showed you for the anterior-posterior axis, that he was right, that actually vertebrates and almost all other animals use the same set of genes to pattern their front-to-back axis, their back-to-belly axis. But vertebrates have indeed reversed the orientation. So what specifies the middle of the belly, the ventral side of a fruit fly, is the same family of molecules that specify the middle of our back, where our nerve cord develops. It does have a slight complication. You have to propose that our mouth is now not in the position that it was when we first evolved. And there's lots and lots of wonderful argument about that. But Geoffroy Saint-Hilaire was probably right. Right, I should be stopping very soon. So if so much is the same, if there is so much conservation in the genetic makeup, the set of genes that make proteins, the organization of genes that specify our axes, what is it that is generating the wonderful diversity of animals, in this case arthropods, that we see today? We probably have to change the way we are thinking about genes to approach this. Genes don't just make proteins. That's the biochemist's view of a gene when you're thinking about what happens to the gene downstream when it's transcribed and translated and makes a protein. But there's something that happens upstream, something that happens with the bits of DNA that are not actually making the protein, but are telling the system where and when to make that protein. There are regulatory elements upstream of genes that receive signals. They receive signals from their own cell, they receive signals from neighboring cells, they receive signals from hormones, they may receive signals from light, from all sorts of sources, and they integrate that information to produce an output, which is whether a gene is on or off. This is an idea that a very influential biologist called Eric Davidson at Caltech has promoted, that we really ought to be looking at genes as much as a microprocessor as we are thinking of it just in terms of part of a machine tool. And if you think of genes as microprocessors, then the way genes talk to one another, the way they interact, is really a network. It is like a circuit diagram in the sense that the activity of one gene will affect another component in the system, will turn it on, will turn it off, will tell a cell that it should be making hair now, and then stop making hair or make it a different color in a little while. And it is these rather essential but complex interactions between genes that are critical for generating diversity. In fact, it is that question of how the interactions between genes change to generate diversity that is the focus of a network of labs, of which my lab is one, studying many of the animals I've talked about and more, and trying to understand whether we can read in genomes the set of interactions between genes that control the network. And the problem is we don't know the code yet. We cannot simply take a computer and say, tell me which genes this particular protein will control. There are techniques that are beginning to figure that out. In fact, this bottom diagram comes from the work of Eileen Furlong's lab, where she is trying to find all the sites in the genome where the controlling protein made by one particular gene is able to interact and bind and do something. But computers cannot do that yet. They are not very good at reading that code. So it's a real challenge for the genome biologists to understand not just what the building blocks are that genomes encode, but what the logic is that genomes encode. My own particular interest is segmentation, is making bodies in repeated units. That's why I'm interested in centipedes. They're very good at making segments. And segmentation is something that is seen in many different animal groups, some of the most successful. And I'm interested in knowing whether it evolved once or at the beginning of animal history or independently several times, whether similar machinery is used, and indeed how, when you have an ancestral machinery that was good at making segments rather slowly, as in the centipede, it evolved to make a fruit fly that can grow from embryo to adult before the banana has dried up. And I'm not going to spend time talking about what we're doing with flies. We understand a great deal about gene networks in flies, not enough, but a great deal. I spent 20 years of my career looking at some of these gene networks. What we'd love to do is to understand how those gene networks have been made different to build segments in this most beautiful embryo. I was told many years ago that developmental biologists start out by looking at their animal and saying, I'm going to understand this. And as they come close to the end of their career, they look at their embryos and they say, aren't they wonderful? And maybe I'm getting close to that stage of my career now, but I still think there's interesting things to do by way of understanding how genomes make animals. Thank you.
In my experience, children have a rather embarrassing way of commenting on how peculiar their relatives are. They don't know half of it, do they? Sponges, sea squirts. And this weekend, when I splodge out into the garden to do a bit of gardening, I really ought to treat the weeds with a bit more respect for the fact they've got more genes than me, and that the wretched snails are much better organized than I am. As a sort of economist, I find it really fascinating that cell development appears to function on very Smithian lines of specialization, of sort of pin factory specialization. Michael, that was a lovely lecture. You've not only given us a wonderful impression of what we do know about the development of species and the development of life, but also very good insights into why we know it. And it's a wonderful start to our series. Thank you very much.